So what we've done in the next sort of 4-minute movie clip is to allow you to see how we, as biologists, try to build a brain and try to, in 3D, take a real brain. This happens to be a brain from a rat, in which we've done some experiments on and tried to understand how these synapses change their structure as the animal learns something. And this is from the hippocampus, a brain region that we know. If you lose the hippocampus, you will not be able to get another new memory. All your old ones will be intact, your new ones will not happen. So as we go through the cruise, this neuropil, you're going to see a central dendrite. The postsynaptic density in red is where those synapses occur, and there'll be some organelles, a mitochondrion will come up in purple, and we'll see some others too, and the axons will be in green, the spines will be in yellow, and the synapses will come up in sort of a copper color. And then we will have the structure that we'll spend more time on later, the astroglia will come in to dance with the rest of this neuropil. So we chose this volume to be a volume that was sufficient to surround this dendrite at the reach of its longest spine. So the first question one can ask is, gee, you have this dendrite, has all these little spines sticking out, how many synapses can it make? And of those synapses, of those axons that come within reach of those spines, what fraction of them actually make a synapse? So now coming in to dance with this dendrite are all the axons in that little tiny volume of neuropil, and you're going to find out how small it is in a moment, that actually make a synapse on that central dendrite. And two of us, Joe Spacek and I, actually manually traced every one of those through 200 serial sections to identify where the synapses occurred. So coming to join them for a moment here is another third partner in the neuropil, a critical partner, and this is the glia, and they interdigitate also as part of this nice felt that is all communicating to make your brain work at this moment. At this moment, your brain is changing, actually, in fact. Some of those synapses are going away and some of them are getting bigger. Because as a mature person, you can't support all the synapses you could possibly have. So now we've jumped into the way I see this tissue when it comes off of the electron microscope. These are the electron micrographs. We're zooming in and out, and all of those structures are structures that I traced with a computer-assisted program and colorized them, named them. So there's some ground truth for these guys who are going to model the brain to know which structures are next to each other and how they interact with each other. Also inside, we have, and now we're going to get to see them in 3D, we're zooming in on that dendrite, and you're going to see just how complex the substructure of these dendrites and their axons are to support these synapses. So the first, you're seeing now the dendrite, and in red are those synapses on those spines. We've made this translucent so you can see some of the 3D structure inside. So the challenge is not just how many connections there are, but how are they supported by these structures that build those synapses. So there's 3 mitochondria that are down the central shaft of that dendrite. And those little white spheres represent the places where protein synthesis was occurring at the time that this tissue was fixed and made. They're called polyribosomes. The blue structures coming in are the cytoskeleton that provides support and the railroad along which all these organelles and proteins get transported. And then now this network, this spider-like network of smooth endoplasmic reticulum is what provides support for regulating ions like calcium, critical ions in the brain, regulating them and also allowing other membrane-bound proteins to move up and down that dendrite to the right place. So we saw a little bit of that felt work neuropil. We saw the dendrite and its axons. Now coming to join the dance are all the other dendrites that surrounded that same central dendrite that we thought was so important, right? Well, these guys are also competing for the axons. And those few axons that I showed you before only synapsed with that central dendrite. Now, the rest of these axons were within reach, but didn't make it. Didn't make a synapse with the central one, but they're synapsing with these other dendrites that are in this volume. And so it's this kind of knowledge that we need to have before we can really have a realistic idea of exactly how your brain is working. And as we go through this, I'm going to tell you at the end here that there are 500 synapses in this volume. 500. This volume is the size of a single red blood cell. It is less than 200 cubic microns. So all of that volume of brain that took us years to reconstruct manually using a computer and putting into this value is so tiny that if we were going to map the entire human brain at that level, we'd have to repeat what I just showed you 8 trillion times.
I see myself becoming a different person every once in a while. And certainly, when I look back on who I was in my 20s, I think of being a different person.
So let me ask you this, and I could